As we wrap up the second season of ACR Journals on Air, I wanted to take a moment to thank you, our listeners. Your engagement in this show has been truly invigorating, and I think of you often as I formulate the questions we all want to know the answers to. As this episode airs in December 2024, our producer par excellence, Joe Rojas, is already planning the third season of ACR Journals on Air. While we are taking a short break, rest assured that we will be back to start that third season on January 21st, 2025. In the meantime, enjoy this episode with Dr. Vicky Worth taking a deep dive into her work studying interferon in the skin. And in the new year, keep your ears and eyes open for Season 3, which will begin with Dr. Mariana Kaplan exploring the complicated web of netosis and why it may be the missing link connecting autoimmune diseases and cardiovascular disease. From all of us here at ACR Journals on Air, we hope you enjoy this episode. Wherever you are in the world and whichever holidays you celebrate, we hope you have a wonderful holiday season and a very happy new year. Hello, and welcome to the ACR Journals on Air podcast. I'm your host, Vicky Shanmugam. Many autoimmune diseases start with a rash, and the skin can be a window into the workings of the immune system. The manuscript we will discuss today is Understanding the Role of Type 1 Interferon in Cutaneous Lupus and Dermatomyositis Towards Better Therapeutics. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. It is a great pleasure to be joined today by the senior author of this manuscript, Dr. Victoria Worth. Dr. Worth is Chief of Dermatology at the Corporal Michael J. Crescenz Department of Veterans Affairs Medical Center and a professor of dermatology at the University of Pennsylvania in Philadelphia. Her laboratory focuses on the effects of UV light on the skin, focusing on the role of inflammatory cytokines and modulation in photosensitivity and photodamage. She's interested in translational work in autoimmune skin diseases, particularly cutaneous lupus erythematosus and dermatomyositis. Dr. Worth is a physician scientist with an active clinical practice and clinical research program through the University of Pennsylvania Department of Dermatology and sees patients with autoimmune skin diseases at the Philadelphia VA. She's also a dedicated educator and established a combined internal medicine dermatology residency program. She has trained nearly 100 fellows, many now practicing worldwide, and she participates in 2 T32 training programs focused on translational dermatology and rheumatology. Dr. Worth is also part of the Lupus Clinical Investigators Network, or LUCIN, which is run through Lupus Therapeutics, the clinical research affiliate of the Lupus Research Alliance. Welcome to the show, Victoria.
It's great to be here. Thank you very much.
So this paper is a review article, and you introduce the topic with a case of amyopathic dermatomyositis. But the real crux of your article is the phenotypic and immune similarities and differences between dermatomyositis and cutaneous lupus erythematosus. And you illustrate this in figure 1 with some incredible photographs, which are well worth a look. I wondered if you could describe those for our listeners.
Surely. So there are different interferons that can be involved in photosensitivity in lupus and dermatomyositis. And what we've pictured are widespread erythematous diviolaceous papules and plaques on the extensor surfaces of the hands. And this is otherwise known as Gotrin's macules or papules. And we see this in dermatomyositis, and it's really quite pathognomonic. It would be very unusual to see this in lupus. And then there's a heliotrope sign, which is erythema and sometimes edema of the upper eyelid. Then there's poikilodermatous erythema on the anterior neck and the V distribution. And again, Gotrin's papules over joints. And so these are all really important to look at. They're very dramatic, and they really help to define what the patient has. The other pictures are showing more cutaneous lupus, and there's psoriasiform subacute cutaneous lupus. There's a patient with malar erythema of acute lupus, which typically does spare the nasolabial fold, annular erythematous plaques of SCLE, and then on the dorsal hands, erythema that's, as opposed to dermatomyositis, where there's targeting of the joints, there's sparing of the joints, and this is characteristic of lupus.
And I wondered whether you could also walk us through figure 2. Your manuscript has some incredible figures, and figure 2 is this beautiful illustration of interferon signaling. Can you turn it into words for our listeners?
Figure 2 shows that type 1 interferons are typically induced by triggers such as viruses, bacteria, or microbial nucleic acids. And these can be sensed by pattern recognition receptors localized in the cytosol or in the endosome of cells. These pattern recognition receptors include toll-like receptors, RIG1-like receptors, and also NLRs, which are known as nucleotide oligomerization domain-like receptors. The type 1 family of cytokines is composed of 17 members. 13 of them are interferon alpha. There's interferon beta, epsilon, kappa. And these bind to the same ubiquitously expressed type 1 interferon receptor, namely IFNAR. And IFNAR is composed of 2 polypeptide chains of IFNAR1 and IFNAR2. The subsequent signaling pathway involves activation of JAK1 and TYK2 and formation of the interferon-stimulated gene factor 3 complex, otherwise known as IGSF3. This includes signal transducer and activator of transcription STAT1, STAT2, and ERF9. This IGSF3 binds to interferon-stimulated response elements and promoters of interferon-regulated genes. So there's a whole cascade of things that go on in cutaneous lupus and dermato, and to some extent, there's more of a targeting of interferon alpha, or more induction of interferon alpha over beta in the cutaneous lupus, and there's more beta over alpha in dermatomyositis. So although they're both often elevated type 1 interferons, there is some difference when you look at a number of patients in terms of which type 1 interferon is upregulated.
And so tell us about UV light as a trigger for interferon signaling in the skin. It's definitely one of the more distressing symptoms for our patients. Why does it happen?
So we know from a number of studies that when UV light shines on the top layer of skin where the keratinocytes are, they have a more robust type 1 interferon response after UV exposure when you compare that to healthy controls. And the pro-inflammatory effect of UVB in patients with SLE is linked to photosensitivity, which is likely stemming from the interferon-rich environment. And there's been a number of labs that have shown that UV stimulation likely upregulates, again, interferon production through endogenous nucleic acid triggering cyclic GMP AMP synthase, which is called CGAS, and also a stimulator of the STING pathway. And this is work that was done by Dr. Elkan and Skopje Gardner. And this is supported by a study showing that when you irradiate murine skin with UV, you get rapid induction of, actually in this case, interferon beta that is blocked by the absence of CGAS. And they had a murine model for this. UVB also has been noted to induce activation of human endogenous retroviruses, which can lead to production of double-stranded RNA, activation of RIG1, and also MDA5, which mediates type 1 interferon induction. So interestingly, the presence of enhanced interferon genes in the skin has been translated to systemic increases in interferon-regulated genes, suggesting that systemic interferon responses can result from sun exposure. UVB can also increase interferon-stimulating capacity of nucleic acid because cells exposed to UV-treated DNA have increased interferon production compared with cells not exposed to UVB-treated DNA. When you inject DNA into lupus-prone mice in the presence of UV light, they can induce CLE-like skin lesions. And if you delete the enzyme that removes degradation of UV-treated DNA, it leads to CLE-like lesions with enhanced interferon production in a STING-dependent manner. So these types of studies really do begin to define somewhat the pathways that are involved for this increased interferon and its effects on the skin. There's also, together, these data suggest that UV modulation of nucleic acids and nucleic acid sensing is critical for UV-mediated interferon responses. In addition, Dr. Kallenberg at the University of Michigan has shown that there is enhanced sensitivity to UV light exhibited by lupus patients resulting from type 1 interferon priming of keratinocytes that drives IRF1 expression and results in caspase 8 activation. And this can lead to increased apoptosis after minimal exposure to UVB. And this is yet another way that we can upregulate the release of intracellular DNA, RNA, and part of that whole cascade. Similar to CLE, cutaneous signs of DM are frequently present on sun-exposed areas such as the upper neck and back and or shoulders. Photosensitivity may be an initial presenting sign of dermatomyositis and should prompt consideration of that diagnosis. And it's really important to differentiate lupus from dermatomyositis because about half the patients I see with dermato come in with a different diagnosis.
Mm-hmm. Yeah, I think we've all seen that mistake happen for sure. We know there are also other triggers for cutaneous lupus and dermatomyositis, and particularly some medications. The TNF inhibitors are well known, but more recently, the checkpoint inhibitors, which are increasingly used as cancer immunotherapy, have also been implicated. Why do these drugs cause immune manifestations in the skin?
Right. So TNF inhibitors have been implicated in both lupus and dermatomyositis onset or flaring. And this may be explained by the fact that TNF inhibition paradoxically increases the production of type 1 interferon. And this can activate, again, antigen-presenting cells and stimulate autoantigen production. And supporting this is an open pilot study of infliximab in patients with refractory inflammatory myopathies, where several patients experienced acute worsening, and these flares were associated with increased type 1 interferon activity in both the blood and the muscle. So in the case of immune checkpoint inhibitors, which target programmed cell death, PD-1, there's programmed cell death, PD-L1 also, myeloid disease and CTLA-4 are increasingly being used to treat both solid and hematologic malignancies by harnessing the immune system's ability to recognize and destroy malignant cells. And these immune checkpoint inhibitors result in a wide range of autoimmune skin manifestations, including lupus and dermatomyositis, as a consequence of basically activating the immune system. The mechanism underlying this is complex, but likely in part due to enhanced CD4 and CD8 T cell activation with subsequent release of cytokines such as type 1 interferons.
So a few supplements can also precipitate similar skin reactions. What are they, and what more probing questions should we be asking when we as rheumatologists take our initial history?
Yeah, that's a great question. We've done quite a few studies now, really sparked because we saw individual cases where somebody would start taking an immunostimulatory herb, and they've been rapid onset, they would get dermatomyositis or have a flare of their disease. And we have now expanded our questioning of patients to a large extent because of these index cases. What we find is very often just asking people, do you take supplements, gets you no answers. They say no. And so we actually now have a handout that we discuss with patients, and we say, okay, these are the herbs we worry about. And they include, and I'll give you a list now, things like spirulina, green algae, chlorella, echinacea, and elderberry, ashwagandha, and alfalfa. There's actually wonderful in vitro data that suggests that these activate the immune system. And in the case of spirulina, we've done a lot of studies to show it activates through TOL4. And so when we take our histories, we ask about these, but then we also show them the names of products where these can be found. For instance, Robitussin now can sometimes have elderberry because, it's thought to boost your protection against viral infections. And actually, the week before the lockdown for COVID, it was a 400% increase in use of elderberry. And ashwagandha is also, for instance, added sometimes to melatonin because it's an anxiolytic and people are trying to sleep. And so these kinds of herbs get added to many substances and many foods. And you have to really assiduously look at labels to know what's in the products. But these things stimulate the immune system, and treatment with them has been associated with a number of autoimmune skin diseases, both onset and exacerbations. We found this to be particularly true with dermatomyositis relative to other autoimmune diseases. And we've published several manuscripts about this. We did some in vitro testing also of another weight loss drug called isoline, which we had a number of patients who started taking it and then had dermatomyositis. It turns out that in vitro, when you dissolve isoline and expose it to peripheral blood mononuclear cells from patients with dermato, they make large amounts of TNF alpha, interferon alpha, and interferon beta, primarily via toll-like receptor 4 activation. We've done similar studies with spirulina and again found the same pathway is activated. And what's really interesting is when you take blood cells from dermato patients and you take them from healthy controls, the amount of stimulation that you see with spirulina is completely different. There's much more pro-inflammatory cytokines. So there's a type of priming that's going on in the peripheral blood cells that really does stimulate the immune system. So I think practitioners should be aware of the potential effects of these herbs on dermatomyositis and ask their patients, as I said, in great detail, not just did you take them, but really go through them in detail. And we do find this handout to be very helpful to elicit a positive history.
Yeah, really helpful information. You mentioned in that mix the known triggering of autoimmunity by viral infections. And I'm wondering, we've also had a number of new vaccines coming out recently, and can some of these reactions also be triggered by vaccines?
So, in genetically susceptible individuals, we know viral infections have been suspected to induce or exacerbate autoimmune skin diseases, and that would include lupus and dermato, through upregulation of type 1 interferons. And recent reports have shown an increased incidence of these disease flares in patients with autoimmune skin disease after getting both COVID-19, but also COVID-19 vaccine, with increasing proportion of patients with dermato compared to patients, for instance, with lupus, having a response which is usually an exacerbation, although occasionally new onset disease. And of the patients who had autoimmune exacerbations after vaccination, 20% had symptoms after the first dose, but most of the patients, it was 82%, had reactions after the second dose and only 4% after the third dose. And so we did see quite a number of patients with mostly exacerbations, most of them manageable, but clearly happening. And we were only able to collect that data prospectively because we saw this happening early on and collected information on a large number of autoimmune patients. Retrospectively, we found patients completely forgot about the association. So again, emphasizing the importance of prospective studies. The underlying mechanisms of immune stimulation are believed to be in part driven by type 1 interferon production, and the COVID-19 vaccines have been shown to upregulate this system. It is unclear why dermato appears to be disproportionately affected. However, differences in cells and type 1 interferon, as we've already discussed, or potentially an increased propensity to flare at any given time may be contributing factors. COVID infection, as I said, similar effects on increasing interferons and inducing flares of disease.
Yeah, really helpful to understand that. What do we know about the role of interferon specifically in cutaneous lupus erythematosus?
A very good question. It's been well documented that CLE has a strong type 1 interferon signature. Histologically, if you look in the skin, in normal skin in patients with lupus, in normal skin produce type 1 interferon with the increased concentration of type 1 interferon response, even if you don't have infiltrating cells. In a recent study using single-cell RNA sequencing of paired lesional, non-lesional skin samples from patients with lupus, there was an interferon-rich signature seen in both lesional and non-lesional keratinocytes, as well as in other cell populations such as fibroblasts and T cells. Pro-inflammatory cellular communication network between, for instance, the stromal cells in the stroma and the skin and inflammatory cells was seen in lesional and non-lesional skin that may support the role of keratinocytes priming myeloid recruitment and inflammatory responses. Further investigation into transcriptional interferon dominant phenotypes and communication networks may provide targeted therapeutic strategies. Other cell populations besides keratinocytes likely contribute to enhanced interferon production in the skin. Most research on type 1 interferon has focused on plasmacytoid dendritic cells in otherwise healthy individuals. PDCs, or these plasmacytoid dendritic cells, produce large amounts of interferon upon recognition of viral antigens, bile, Toll7 and Toll9. However, the role of PDCs in type 1 interferon production in autoimmune disease like SLE is less clear. There was one study by Steros et al. Showing that in preclinical autoimmunity and in SLE, PDCs are not the effector cells, but have rather lost their capacity for toll-mediated interferon alpha and TNF production, failed to induce T cell activation independently of disease activity and blood interferon signature. PDCs in lesional CLE, we found in one of our studies, were not major type 1 interferon producers. This may be in part due to the inflammatory environments with high TNF production, such as can be found in the skin of patients with lupus. Additionally, monocytes exhibit sting-dependent type 1 interferon production after UVB stimulation. How the inflammatory environment modulates the functional status of PDCs and other interferon-producing cells in lupus is still being researched.
And what about dermatomyositis? What are the data telling us there about interferon?
So we know that overexpression of type 1 interferon has been linked to the immunopathogenesis of dermatomyositis. Interferon-inducible transcripts and activated type 1 interferon signatures are observed in patients with dermatomyositis. There's increased expression of interferon alpha beta inducible genes and proteins that are induced by type 1 interferon, such as the MIX-A protein, seen in the peripheral blood mononuclear cells and also in muscle and skin of patients with. There's elevated blood interferon beta protein seen in dermatomuscle and blood, and this correlates with the blood type 1 interferon inducible gene, and the interferon beta transcript can readily be found in the skin and DM. This protein is highly upregulated in T cells, macrophages, dendritic cells, and endothelial cell populations. Additionally, interferon signatures, in particular interferon beta, closely correlate with skin disease activity. And there have been a number of studies that looked both in the sera and in the skin that have shown correlation with the amount of skin activity. Interferon beta expression is abundant in all cells present in the epidermis and dermis of dermatoskin. TOL3 is elevated in vascular endothelial cells, myeloid dendritic cells, and regenerating myofibers in patients with juvenile dermato. So I think that interferons are very important in both lupus and dermato, but there are some nuances that really will, I think, be important to understand as we get to more directed specific therapies.
Oh, yeah, no, that's a great lead in. And it is a wonderful time in the field of both dermatology and rheumatology because we do have some emerging therapies. And I wanted to have you walk us through some of the clinical trial data on some of those emerging therapies, specifically those targeting type 1 interferon and the downstream pathways for cutaneous lupus erythematosus and dermatomyositis. I'm going to go through these in the order that you present them in the paper. So I wondered if you could start with cifalimumab. This is an inhibitor of interferon alpha. Does it have impact in dermatomyositis?
So there was a phase 1b clinical trial study observing that anti-interferon alpha, the cifalimumab, inhibited type 1 interferon signatures in patients with dermatomyositis. It's very specific for interferon alpha and leaves other type 1 interferons unaffected, and the skin is able to bind to IFNAR from these other type 1 interferons. So there was no real clinical data. It was more at a signature level that there seemed to be some impact. And this is as opposed to in lupus, where there was clearly studies that showed that the drug worked, but it wasn't as effective as some of the other drugs that we'll be talking about, likely because, again, it's more specifically blocking certain types of interferon alpha. Given that the current data for dermato seems to be predominantly driven by interferon beta compared with interferon alpha, it really is unclear whether further investigations into interferon alpha inhibition will yield efficacy in treating dermato skin disease.
And the next agent is dazukibart. It's an interferon. What does it do and how does it impact skin involvement?
There was a phase 2 trial of this drug. It's a basically anti-interferon beta, which is an immunoglobulin G1 neutralizing antibody. It was tested in adults with dermatomyositis, initially in patients who had skin disease, and then subsequently patients who had muscle disease, and many had skin at the same time. The clinical endpoint for the trial in skin dermato was the CDASI, the Cutaneous Dermato Disease Area and Severity Index. And we know what is a significant improvement in that validated measure, and a reduction of less than 5 points was achieved in 100% and 96% for either the low dose or high dose of the drug. The placebo response was about 35% for this trial. And for the CDASI, reduction of greater than 40%, the rates were 80, which we know is, by the way, significant from a patient perspective, a 40% improvement in the CDASI. The rates of improvement were 80%, 82% for the 2 doses of the drug versus 7% for the placebo. So clearly a huge difference between the treated and placebo-treated arms. A smaller muscle trial trended to improvement as well in the treatment improvement score. There's now an ongoing phase 3 trial that's studying predominantly or only patients who are weak and have dermatomyositis.
And next is a drug that we've talked about before on this podcast, anifrolumab. Can you remind us how it works and then what the clinical trials show for this drug?
Yes, so anifrolumab is an anti-IFNAR1 monoclonal antibody, which shows significant effects in decreasing disease activity of SLE in a phase 2 trials and another phase 2b study in patients with moderate to severe SLE that was considered very successful. There were two phase 3 trials, TULIP 1 and TULIP 2, and TULIP showed also significant efficacy that led to FDA approval of this drug for SLE. And this was completed in 2019. And we know it showed promise in treatment of skin disease and lupus with a greater than 50% reduction in CLASI scores noted at week 12 in patients with a baseline CLASI score greater than 10. And that was significant relative to the placebo group. Several recent case series have shown promise of anifrolumab for the treatment of refractory CLE. There's an ongoing study for CLE, but right now it's not FDA approved for that indication, and so it's much harder to get that drug for patients who only have CLE. IFNAR blockade blunts the effects of both interferon alpha and interferon beta and has been shown to reverse many of the proteomic and cellular changes of enhanced type 1 interferon that are seen in SLE. And given that there are similar interferon-driven changes in dermatomyositis, further studies are warranted to determine if IFNAR blockade may provide clinical benefit, and those are ongoing.
Perfect. So you also mentioned earlier the important role of the plasmacytoid dendritic cells, and it is possible to block not the interferon itself, but the plasmacytoid dendritic cells that are implicated in interferon production. Filimab is a monoclonal antibody that binds to circulating dendritic cells and inhibits production of type 1 interferon. Does it show benefit in either dermatomyositis or CLE?
Okay, well, this is an interesting drug. And what it does is an antibody against BDCA2, and its binding leads to internalization of the receptor and downregulation of type 1 interferons, TNF, and other pro-inflammatory cytokines and chemokines. There's no data in dermato. The cells in dermato skin are more frequently myeloid dendritic cells as opposed to plasmacytoid dendritic cells. So it's unclear if that approach will work, but it will be interesting to test it. Litifilimab is a monoclonal antibody that binds to blood dendritic cell antigen 2, as I said, and it's been found to be beneficial to patients with both SLE and CLE. In fact, both of those phase 2 trials were published in the New England Journal. PDCs have been implicated dermato with increased numbers noted in DM skin, but not nearly to the extent of CLE. And when we look at DM skin, again, we see mostly myeloid cells. Single-cell analysis using mass cytometry of DM skin supports some increased PDC numbers in DM skin, again, but we don't think that's going to end up being the main driver in DM. What I would basically say is that this is a very interesting therapeutic approach for cutaneous lupus and SLE, and there are phase 3 trials that are now ongoing, pretty far along, and stay tuned. Maybe by the next time we have a review, we'll have some results from that. But the phase 2 trial is very encouraging.
Yeah, really, really exciting work and reminds us the crucial importance that not all photosensitive rashes are the same at a molecular level. So it does matter to define the disease phenotype correctly. So we've covered all the new monoclonals. But what about the rheumatologist's friend, hydroxychloroquine? We use it a lot in lupus and very commonly in people who are photosensitive. So how does it work? And why does hydroxychloroquine sometimes not work and even sometimes cause a flare in people with dermatomyositis?
Yeah, that's a great question. I don't know that we have the 100% answer for that, but we do know that hydroxychloroquine binds to nucleic acids and the activation of toll-like receptors, which are cellular receptors that trigger inflammatory responses. And this prevents activation of toll receptors, which are key to the production of TNF alpha, as well as interferon-regulated genes. Hydroxychloroquine also accumulates in lysosomes and autophagosomes and increases their pH and is thought to have some alterations there as well in terms of protein degradation. Plaquenil or hydroxychloroquine interferes with antigen processing in macrophages and other cells that activate antigens, and it also inhibits autophagy by blocking the fusion of autophagosomes and lysosomes, which prevents recycling of metabolites and results in an accumulation of autophagosomes. The balance between innate immunity and adaptive immune responses needs to be considered when considering autophagy as a therapeutic target, and it's possible that the balance is a little off in dermatomyositis for some patients, and that it's more balanced towards immune stimulation due to inhibition of the immune system with autophagy. There's a lot of work to be done to better understand that. We know that about 20% of patients who get hydroxychloroquine when they have dermato can flare, but another 20-25% of the patients who can take it do respond, and that is very helpful so they don't have to go on to more immunosuppressive medications.
Yeah, so it's really a case of kind of suck it and see, which is tricky, I think, for both parties, the physician and the patient.
The other thing I wanted to mention is that the dominant cytokine profile in inflammatory cell populations of CLE lesions are really quite more heterogeneous than we totally realize. And this could also reflect a different responsiveness to antimalarials. So, for instance, hydroxychloroquine responsive patients have significantly higher type 1 interferon regulated transcripts and lower TNF levels than those who are refractory to hydroxychloroquine. And in contrast, patients who are refractory to hydroxychloroquine, who, for instance, we might be adding quinacrine, which is another medication that we sometimes add, had significantly upregulated TNF expression produced by myeloid dendritic cells. So some very differences in the types of cells and actually the cytokines they're producing. Toll-8 is a sensor that detects viral and bacterial pathogens and is expressed on the endosomal membrane of cells like monocytes and macrophages, which are really important in the skin disease of dermato, but they're not on PDCs. And so it really may be that having different cell types makes a difference in their ability to respond to hydroxychloroquine.
Yeah, I mean, I think that's going to be so important in the future when, we hope that we might get to a place where targeted therapy is possible and perhaps, it's actually feasible to consider that a skin biopsy might actually allow physicians to know the answers to these questions ahead of treatment. So let's move on now to the JAK inhibitors. They act downstream of the interferon receptor, so it seems like they should be good sites to target. There have been some positive studies. So where are we with clinical trials of JAK inhibitors for CLE and dermatomyositis?
So this is really a more targeted downstream approach, and it's certainly been tried. We know that inhibition of JAK-STAT pathway, which is important for signaling of inflammatory cytokines, immune regulation, and different cell growth factors, would be a prime target. Baricitinib was a drug that was studied for SLE, and it's a selective inhibitor of JAK1. In early trials, it showed efficacy in SLE, but failed in 2 phase 3 trials, unfortunately. And we could go into all kinds of details with that. I mean, the amount of skin disease in those trials was relatively minor. And so it's a little bit hard to know if it might have been effective if it had been more of a CLE trial. Tofacitinib is an inhibitor of JAK1 and 3, is approved by the FDA for a number of autoimmune problems, but there's been limited reports of it working, for instance, in recalcitrant cutaneous lupus and as well as in dermatomyositis. But we don't really have placebo-controlled trials yet to be able to know for sure. In a recent phase 1 double-masked randomized safety trial of tofa, it was suggested that inhibition of interferon signaling results in improvement of certain cardiometabolic and immunologic parameters associated with, for instance, premature athero and SLE, which is a little bit different than what we hear about with the JAK inhibitors. And so I think we have a lot to learn about this drug. Clinical trials of tofa and SLE and CLE are still progressing. There was a study with filgotinib, a highly selective JAK1 inhibitor that failed in a phase 2 trial. And a few studies have reported a positive response to JAK inhibitors, as I said, in dermatomyositis. In particular, one was a 10-person open-label trial, and it seemed to work pretty well for the skin. One of the problems with this studying dermato is that often these patients are on background medications, which also continue to work within the trial, and so the placebo responses can actually be fairly dramatic. And so I think that we have a lot yet to learn about this approach of JAK1 and TYK2 kinds of inhibitions, but we'll get to TYK2 next, I think.
Yeah, okay. Well, you led us right in. So the newer TYK2 inhibitor, deucravacitinib, is now approved for people with moderately severe plaque psoriasis. You've mentioned that there are some psoriaform lesions sometimes in CLE. So does this agent show benefit for CLE or dermatomyositis?
Yeah, so deucravacitinib is an oral selective allosteric inhibitor of TYK2. It binds to regulatory domain and locks the enzyme in an inactive state. And so it's distinguished from inhibitors of JAK1, JAK2, and JAK3 that bind to highly conserved active domains. And so deucravacitinib is FDA approved, as you said, for psoriasis. And there was a recent phase 2 randomized double placebo-controlled trial suggesting that there was a much higher response rate for CLASI 50 in 3 doses relative to placebo. And this was very exciting. These patients all had SLE, and there's an ongoing SLE phase 3 trial Unfortunately, it would be wonderful to get some results for CLE as well, because it can be difficult to access drugs if we don't get separate approvals for CLE.
Yeah, no, it's a perpetual problem, I think, with some of these rarer autoimmune diseases. So the last group of drugs that I wanted to talk about is the drugs that impact the endocannabinoid system. So we know this can modulate inflammatory pathways, and the drug in question is lenabasum, which is a selective CB2 agonist. What do the clinical trials for this agent show?
So there was a phase 2 randomized placebo-controlled clinical study, actually single site at my institution, and it showed that linabasum treatment was well tolerated and associated with improvement of dermatoskin using the CDASI, as well as other multiple efficacy outcomes in patients with moderate to severe skin involvement. Additionally, linabasum had a significant effect on the interferon beta gene signature in the skin, and it also reduced IL-31, which is an itch protein in the skin that can be significantly mediating itch, which we know is a huge problem for dermatomyositis. So the degree and consistency of clinical benefit combined with the favorable safety profile led to a phase 3 trial. But unfortunately, because of some FDA issues, the pivot was to more muscle patients and an endpoint that is pretty insensitive to improvement in skin. That trial failed. However, the 10% of patients who had clinically amyopathic dermato showed really the same response that was seen in the phase 2 trial. So the mechanism is really interesting and perhaps differentiates some responses between classic and amyopathic dermatomyositis patients.
Really, really interesting. So that was quite the tour of new therapies, but you wrap up your review with a pretty old treatment that we know now is effective in myositis, which is IVIG. Would you mind telling us how does it work and why did you as a clinician really want to use IVIG in the case that you presented?
So one of the actions of IVIG is to block the FcRn receptor, and this basically helps to clear pathogenic antibodies. And so it's the mechanism that we look for. We know that many patients with dermatomyositis, but I will tell you not all, have antibodies that are specific for myositis, and potentially those are cleared as a result of blocking FcRn with IVIG. But there are likely other mechanisms as well for its efficacy. There was a prospective study of IVIG in dermatomyositis and demonstrated efficacy and led to FDA approval of IVIG for that indication. We know from that study that the patients who had skin disease, which was most of them, also had improvement. And it's certainly a drug that we use for patients that have unresponsive disease to other modalities, such as hydroxychloroquine or immunosuppressives. And so it is definitely an option that we find very useful. In addition, for patients who have perineoplastic dermatomyositis, where we may not want to immunosuppress them further. So it's effective therapy. Not every patient tolerates it, but it can be very helpful.
Yeah, really exciting times. And I want to thank you for bringing us up to speed interferon in the skin and the evolving world of therapeutics. I want to spend a few minutes now just learning a little bit about what drives you as a physician and as a researcher. You trained first as a dermatologist, but unlike many dermatologists, your focus goes deeper than the skin. What drew you into immune dermatology and the field specifically of lupus and dermatomyositis?
Great question. So actually, I was an internist first and then subsequently did my dermatology residency. And I really felt that derm was a subspecialty of internal medicine. Many people that originally went into dermatology many years ago were internists first and then became dermatologists. So it became apparent to me when I joined the field that there were a large group of patients that were sort of falling between the cracks, where they didn't have systemic disease, but there also weren't really necessarily dermatologists in a particular community that were comfortable taking care of their extensive skin problem and monitoring them for systemic disease. So it was a natural fit for me to be able to use my internal medicine. I'm more holistic in my approach, and to also examine the patients. I also think and as a result of that, we have done a few things. We have a Med Derm Society now. We have a Derm Room Society. So we have a group of people, many of them young, who are coming up in the ranks, who I think will be comfortable with this part of the field. And we also have a combined Med Derm program where people come out double boarded in internal medicine and dermatology. So part of why I was attracted is that there were no approved therapies for like 70 years for dermatomyositis or cutaneous lupus. Our patients really are somewhat desperate, and it's really important to improve the situation. And if you think of things like psoriasis, where there's just enormous numbers of drugs, we're truly lagging behind. And there are lots of reasons for that. But these diseases, that dermato and lupus, have enormous impact on patients. And so it's a great field to really focus on just because it's really interesting clinically and also really interesting translationally and in the lab.
Yeah, I mean, I think sometimes we underestimate the impact actually of these skin rashes on patient quality of life and how it really impacts the patients.
The other thing is that I would say that as I started getting into the field, and did do a fellowship in a lab and laboratory-based fellowship, but also had the benefit of working in a lupus clinic, I realized that to advance the field, we were going to need ways to measure better than what we had, which was there were no trials and there were no measures. So that was an area that basically got my focus initially.
Yeah, well, so that leads me to my next question, which is you've been instrumental in developing several tools for measuring treatment effectiveness on the skin, in particular, a score called the Cutaneous Lupus Disease Area and Severity Index, or CLASI, which, if I may say, is a very classy name. What drove you to develop this tool, and how are you recommending people use it?
Sure. So as I started to think about doing clinical studies and translational studies, I realized that there was a big impediment with the lack of a validated tool to evaluate disease severity. So I was interested in, again, looking at getting new therapies, but there was no way anybody was going to study this disease without a validated tool. And so that led me to the CLASI, which we initially published in 2005. Here we are almost 2 decades later, and it's been an enormous effort with many studies, validation studies, biomarker studies. It's been an interesting road dealing with the FDA, which never did studies in this disease. And it's not great to do studies in a disease that's never been studied, because you really have to do a lot of education and a lot of work to get to the point where you have endpoints that will be able to be used for studies.
Yeah, so laying the groundwork is crucial. The CLASI is also one of 4 projects being advanced by the Lupus Accelerating Breakthroughs Consortium, or Lupus ABC project. This is a public-private partnership bringing together individuals living with lupus, their advocates, industry, clinicians, researchers, the US government, including the FDA, and really focusing on accelerating development of new treatments for people living with lupus. Tell us about the Lupus ABC project and then a little bit about your role in that consortium.
Sure. So the Lupus Accelerating Breakthroughs Consortium grew out of a patient-focused drug development session that was held at the FDA several years ago. And this was actually a combined effort between the Lupus Research Alliance and the Lupus Foundation of America, and was a chance for the FDA to hear about the impact of lupus on patients. And that set the groundwork for then being able to form a partnership with the FDA, which is what this consortium is, and to have dialogues to try to advance the ability to study the disease and to understand the disease. So the LRA took the initiative again to follow up on this patient-focused drug development to identify projects that could make a difference in the development of lupus studies. And so the initial consortium was really a partnership between the FDA and LRA. At the initial meeting in April of 2023, there were several projects that were presented. I pitched a project related to working with the FDA to allow use of CLASI as a primary outcome for CLE studies. And up until now, that has not been allowed as a primary outcome, although it has been for phase 2, but for phase 3 registration trials, it has not been allowed. And it's used as a secondary outcome in SLE trials, despite, as I said before, years of validation studies demonstrating reliability, responsiveness, and correlation with quality of life and biomarkers, and successful phase 1 through 3 studies. So it's been, a little bit like hitting your head against a brick wall. And so the Lupus ABC group, being the group that it was, has made great progress with a dedicated group, as you mentioned, academics, industry, patients, all partnering with the FDA. And this has provided the opportunity to inform the FDA about cutaneous lupus and the importance of using a validated disease severity tool to advance the field. And most studies have required use in dermatology of something called an investigator global assessment tool. And that tends to be a little bit subjective. It's like a 5-point tool. And very often it's driven by what is the worst attribute. In general, overall, like assessment of, well, this is how much erythema they have or how much scale. And then one of those things will drive how you rate the patient. And so it turns out that in a rare disease, you would have to have more patients in order to have that kind of a score work in general, unless you have a great drug. And maybe we're going to get to the point of a great drug, but we're not quite there yet. And so we really need to have more granularity in the tools that we have so that we can show that there's a difference between the drug and the placebo. So as a result, there have been a paucity of CLE trials, and it's been challenging for companies interested in developing therapeutics for CLE to have a path forward. And this has delayed or led to termination of a number of trials of promising therapeutics. So this work of the ABC group with LRA is just incredibly important. And I can't think of a better mechanism for trying to address some of these issues.
Yeah, I feel like you've actually really finally started to make some headway. So kudos to you and all of the partners in that program. So it's also essential to improve recruitment to clinical trials and really ensure that diversity of people living with lupus is reflected in the clinical trials that we execute. You've been working with Lupus Therapeutics on a program called Project Change. What is Project Change, and how did you get involved with that program?
These things all fit together, believe it or not, because if you're doing clinical trials, you want to have access to patients. If you're not getting access to the full range of patients, that's a problem in terms of evaluating a drug. So as part of Lupus Therapeutics, the trial group linked to the LRA, this group is really critical to identifying academic sites of committed physicians with expertise in lupus to advance the study of lupus through clinical trials, biomarker studies, and now Project Change. And so there were 3 sites that were selected to have pilots for Project Change, namely Emory, Vanderbilt, and ASA Penn. The purpose is to determine the barriers and possible solutions to a lack of participation on the part of Blacks in clinical trials. And we know that Blacks make up about 43% of the lupus population, but only 14% of lupus clinical trial participants. And response to lupus treatments may vary by race, and Blacks are more likely to have severe disease. So I was interested in improving the care of lupus patients overall, and clinical trials, I think, are critical to that effort. We've just discussed some amazing drugs that are being developed, but we need patients to participate to get the approval for these. In addition, CLE patients have the additional barrier of not being able to access these drugs, as we talked about, because they don't have SLE, and we need to be able to get approval for CLE, but we also need to get people who are Blacks, who are other races, to be in our trials. My goal is to provide, and what we've worked on is we've formed a group in Philadelphia of community-based people ranging from social workers to patients to advocates and other physicians from renal, rheum, and derm at different institutions, and tried to come up with some ideas of how can we advance the participation and understanding why there are these blocks. We are currently doing a community-based survey to better understand the barriers from a lupus patient's perspective, and then hope to develop plans to accomplish goals based on the feedback that we're getting. We work very much with different organizations in town now, and we have people that are lupus warriors with the Lupus Foundation, and we really want to have a sort of group that can try to advance the access and understanding why people are not choosing to do trials. We have a wonderful steering committee, as I said, and it's really been a chance to really get together and talk about how do we approach this? What do we do? And I think we're still working on this. I don't think this is an easy problem. It's something that people have struggled with for a long time. But I do think that there is something to the being community-based and trying to work with people at the level where they are, if they're not in an academic center or even knowledgeable about trials.
Yeah, I love that. And it's, I think, really helpful to hear about these initiatives and about the work you're doing at the kind of grassroots level to try and improve engagement. I want to thank you. This was a wonderful discussion. I've been itching to talk to you for some time, so I was thrilled you could join us today and share all of your expertise.
Well, thank you. It's been great to be able to talk with you.
So today, I also need to extend a thanks to the immune dermatologists who share our passion for immunology and are never scared away by a rash consult. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at AC Rheum on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.